Effect of insulin on fat metabolism. So this occurs via increasing the glucose transport into the liver cells, and this glucose will then via the EMP pathway of glycolysis form the pyruvate, and pyruvate will then enter into the citric acid cycle via the acetyl coenzyme A formation YPDH, and it will form more and more citrate and isocitrate ions. Why? Because citrate and isocitrate ions are more activators of carboxylase of acetyl coenzyme. So acetyl coenzyme A, which is forming malonyl coenzyme A after carboxylation, and this is done by an enzyme acetyl coenzyme A carboxylase, and this is activated by citrate and isocitrate. So this is why these molecules will form in more quantity for synthesis of fat. And most of the fatty acids are then synthesized within the liver and used to form triglycerides. So in this case, glycerol and fatty acid will combine and form the triacylglycerol, and they will be stored in the liver. Role of insulin in storage of fat in adipose tissue. So in adipose tissue, this hormone sensitive lipase enzyme is inhibited, so that removal of fatty acid from the triacylglycerol will be inhibited. So when you have plenty of food, plenty of energy, so triacylglycerol will not remove any fatty acid, and this action is done by hormone sensitive lipase, and this will be inhibited. Right, and insulin promotes glucose transport through cell membrane into the fat cells. So extra glucose will be coming into the fat cells, and it will be utilized for the formation of fatty acid as well as glycerol, and these two will combine along with other fatty acid formed by other pathway to form the triacylglycerol. Insulin deficiency increases use of fat for energy. So when you have no insulin, and what is the effect on fat? So insulin deficiency causes that hormone sensitive lipase to get activated, and that will lead to the lipolysis of storage fat, and free fatty acids will be released into the circulation for the energy, and this will be utilized for by breaking down it into fatty acid will break down into coenzyme A, they will acetyl coenzyme A, and they will be entering into the citric acid cycle to form the various intermediate to provide the energy. Insulin deficiency increases plasma cholesterol and phospholipid concentration. So this it does by when insulin is not there. So your triacylglycerol which is formed, which is forming little bit of phospholipid also by breaking it down and assembling the phospholipid and cholesterol, and these molecules will then combine in the VLDL, and they will be circulated into the blood. So this it does by activating various enzymes required for this process. Excess uses of fat during insulin deficiency cause ketosis and acidosis. How it does? The carnitine transport mechanism for transporting fatty acid into the mitochondria becomes excessively activated in the absence of insulin, and that will lead to the beta oxidation of fatty acid to form acetyl coenzyme A, and this acetyl coenzyme then combine to form acetoacetic acid, and this acetoacetic acid then gets circulated into the blood to be utilized for different tissue. And when excess of acetoacetic acid is formed, this can form break down into beta hydroxybutyric acid and acetone. And when excess of acetoacetic acid is there, this will lead to the formation of acetone and beta hydroxybutyric acid. So these three are called as ketone bodies, and when excess of beta hydroxybutyric acid is there, as it is acid, it will also increase the acidity or decrease the pH, and this condition will lead to the acidosis. So diabetic patient who have lack of insulin, or their cells are sensing the lack of insulin, this kind of condition can occur. Effect of insulin on protein metabolism and growth. So this does by activating different mechanism. Insulin stimulates transport of many of the amino acids into the cells. So these amino acids will be utilized for the formation of different proteins, and specifically it is incorporating valine, isoleucine, leucine, tyrosine, and phenylalanine. These enzymes, these amino acids are transported via the insulin, and this effect is similar to the growth hormone, which is having different amino acid specificity. Insulin increases translation of messenger RNA. So the enzymes which are required for the synthesis or biosynthetic pathways, those the messenger RNA for them will be translated by activation of gene. So this process is mediated via insulin, and over a long period, insulin also increases the rate of transcription of selected DNA genetic sequences. When there is a lot of insulin for longer time, this will lead to the transcription of genetic sequences for which are required to express a specific gene for different enzymes to store the fat, carbohydrate, and amino acid or protein. And at the same time, insulin inhibits the catabolism of protein. So protein at this time when synthetic pathways are activated, so protein catabolism or degradation of protein will be inhibited. In the liver, insulin depresses the rate of gluconeogenesis. As we discussed earlier, that amino acids are involved in the formation of gluconeogenesis. So this will inhibit the incorporation of amino acid into the glucose by the inhibition of glucose amino acid transport into the liver. So this amino acid will be spared from this effect for the formation of glucose. All right. Now, what is the effect of insulin deficiency on protein metabolism? So it will cause protein depletion and increased plasma amino acids. Protein synthesis pathways will be stopped while protein degradation will be increased. So this protein will be utilized for the energy, which is utmost requirement during the starvation period or in the acute or diabetic condition. So this will result in protein wasting. Right? Wasting. Insulin and growth hormone interact synergistically to promote growth. So in this case, one experiment is done in which a rat has the no hypothalamus, or its hypothalamus is removed. No hypothalamus, and as well as no pancreas. Its pancreas is also removed, and then the effect of growth hormone and insulin is seen on the weight of the rat. So it is seen that when you add growth hormone or insulin separately, what happens, there is not much effect on the weight gain or growth of the rat. But when you add both the things together, so it does have effect. It shows exponential increase in the growth of the rat or weight gain of the rat. So it shows that it has like they function synergistically to in the growth of the organism, and it does via the specificity for the different amino acid, like insulin are able to absorb, or insulin are able to increase the transport of some specific amino acid, while growth hormone has specificity for different amino acid.